Neanderthals. Our closest extinct human relatives, the Neanderthals, have long fascinated scientists and the public alike. They roamed Earth for thousands of years, adapting to various environments and climates, yet they vanished around 40,000 years ago. Their disappearance is a puzzle, a mystery that has captivated scientists for generations. What could have led to the extinction of such a resilient species? What happened to these skilled hunters, these masters of fire, these crafters of tools? They were adept at survival, yet something went terribly wrong. The answer, it seems, lies buried deep within their bones, etched into the very fabric of their DNA. Their genetic code holds the secrets to their past. Recent breakthroughs in genetic analysis have provided unprecedented insights into the lives and demise of Neanderthals. These advancements have opened a new window into their world. By extracting and analyzing ancient DNA from fossilized bones and teeth, scientists have begun to unravel the genetic secrets of these enigmatic hominins. Each discovery brings us closer to understanding their story. These studies have revealed a complex interplay of factors that likely contributed to their extinction, with genetic vulnerabilities playing a significant role. It wasn't just one thing, but a combination of challenges. The Neanderthal genome, painstakingly pieced together over years of research, paints a picture of a species grappling with challenges both familiar and unique. Their genetic story is a testament to their struggle. Just like us, Neanderthals faced the capricious whims of nature, the ever-present threat of climate change, and the pressure of competition for resources. Their environment was both a provider and a challenge. However, their genetic makeup suggests that they also contended with a more insidious threat, one that eroded their resilience from within: a lack of genetic diversity. This genetic bottleneck may have been their undoing, making them vulnerable to diseases and reducing their ability to adapt. The study of ancient DNA has revolutionized our understanding of the past. Like fragments of a shattered mirror, scattered across time, these genetic remnants offer tantalizing glimpses into the lives of long-gone creatures. By carefully collecting and analyzing these molecular fossils, scientists can reconstruct evolutionary histories, trace migrations, and even uncover the genetic underpinnings of extinct species traits. In the case of Neanderthals, ancient DNA has been instrumental in revealing the genetic factors that may have contributed to their demise. By comparing Neanderthal DNA with that of modern humans and other ancient hominins, researchers can identify genetic changes unique to Neanderthals, providing clues about their biology, adaptations, and vulnerabilities. One of the most striking findings from ancient DNA analysis is the discovery that Neanderthals possessed relatively low genetic diversity compared to modern humans. This lack of diversity, a potential sign of small population sizes and isolation, may have had profound implications for their ability to adapt to changing environments and challenges. Imagine a bottle filled with raznotsvetny marbles, each marble representing a different gene variant within a population. These marbles symbolize the rich genetic diversity that exists within a healthy population, where each variant plays a crucial role in the overall adaptability and resilience of the species. Now imagine that the bottle is suddenly constricted, allowing only a few marbles to pass through. This constriction represents a significant event that drastically reduces the population size, such as a natural disaster or a sudden environmental change. The marbles that make it through the bottleneck represent the surviving gene pool, while the others are lost. This loss of marbles signifies the loss of genetic variants that did not make it through the event, leading to a reduced genetic diversity. This is a simplified illustration of a genetic bottleneck, a sharp reduction in the genetic diversity of a population due to a dramatic decrease in its size. Such events can have long-lasting effects on the genetic health of the population. Bottlenecks can occur for a variety of reasons, such as natural disasters, disease outbreaks, or migration events. Each of these events can severely limit the number of individuals that survive and reproduce, further reducing genetic diversity. Regardless of the cause, the result is the same: a loss of genetic variation. This reduction can make the population more vulnerable to future challenges, as there are fewer genetic options available to adapt to new threats. Genetic diversity is essential for the long-term health and survival of a species. It ensures that there are enough different traits within the population to cope with changes and challenges in the environment. A diverse gene pool provides a wider range of raw materials for natural selection to act upon, increasing the chances that at least some individuals will possess traits that allow them to survive and reproduce in the face of environmental challenges, diseases, or other threats. This diversity is the key to resilience and adaptability in the natural world. How do we know that Neanderthals experienced genetic bottlenecks? This question has intrigued scientists for decades, leading to numerous studies and research projects aimed at uncovering the mysteries of our ancient relatives. The evidence comes from various sources, including their DNA and even the shape of their bones. By examining these clues, researchers have been able to piece together a more comprehensive picture of Neanderthal life and their genetic history. One study published in the journal focused on the morphology of the inner ear, a structure known to be under tight genetic control. The inner ear, particularly the semicircular canals, plays a crucial role in our sense of balance and spatial orientation. By analyzing the intricate shape of the semicircular canals, tiny fluid-filled tubes within the inner ear that help with balance, researchers found significantly less variation in Neanderthal ears compared to modern humans. This discovery was pivotal in understanding the genetic constraints faced by Neanderthals. This reduced morphological diversity suggests a history of genetic bottlenecks, as these events can limit the range of variation within a population. Genetic bottlenecks occur when a population size is significantly reduced for at least one generation, leading to a loss of genetic diversity. Further evidence of bottlenecks comes from the analysis of Neanderthal DNA itself. By sequencing and comparing the genetic material of different Neanderthal individuals, scientists have been able to identify patterns that point to these bottleneck events. By comparing the genomes of different Neanderthal individuals, scientists have found patterns of genetic variation that are consistent with a history of population bottlenecks. These patterns reveal the challenges Neanderthals faced in maintaining their population size and genetic diversity. These findings suggest that Neanderthals experienced at least one major bottleneck event around 110,000 years ago, significantly reducing their genetic diversity. This event likely had profound effects on their ability to adapt and survive in changing environments. Section 5. Social isolation. A recipe for inbreeding. Genetic bottlenecks are not the only factor that can erode genetic diversity. Social isolation, where populations become geographically or culturally separated, can also lead to a loss of genetic variation through a process called inbreeding. When populations are isolated, individuals have a limited choice of mates, increasing the likelihood of mating between close relatives. While mating between close relatives is not inherently harmful, it can increase the chances of offspring inheriting two copies of recessive genes that carry harmful mutations. These mutations are often masked when paired with a healthy copy of the gene from an unrelated individual. But when two copies are present, they can manifest as genetic disorders, reducing fitness and survival. Inbreeding can have particularly devastating consequences for small, isolated populations. As the frequency of harmful mutations increases, the overall health and reproductive success of the population can decline, making it more vulnerable to extinction. Neanderthals lived in small, isolated groups. This isolation made it difficult for them to maintain genetic diversity. Over time, their genetic pool became more and more limited. Without fresh genetic material, they faced increased risks of genetic disorders. This isolation was a significant factor in their eventual decline. As modern humans began to spread across Europe, competition for resources intensified. Neanderthals, already struggling with their limited genetic diversity, found it increasingly hard to compete. Climate changes also played a role, further stressing their populations. The combination of these factors created a perfect storm, leading to their eventual extinction. Genetic diversity is crucial for the survival of any species. It allows populations to adapt to changing environments and resist diseases. Unfortunately, Neanderthals lacked this diversity. In contrast, modern humans had a more varied genetic pool, giving them a significant advantage. This diversity helped modern humans to thrive while Neanderthals dwindled. Despite their extinction, Neanderthals have not been entirely lost to history. Modern humans carry traces of Neanderthal DNA, a testament to our shared ancestry. These genetic echoes influence various aspects of our biology, from immune responses to skin pigmentation. In many ways, the story of Neanderthals continues to live within us.